Bismillahirrahmanirrahim. This is me, Asma Kamal Thebu, and today I'm going to presenting molecular structure of cell organelles. Before understanding the molecular structure of organelles, we have to know about the cell. That what is a cell? As we know that cell is the basic structural and functional unit of life. Whatever the organism, either archaeobacteria up to the blue whale, all organisms are made up of cells. Cell. Now question arises that what's found inside a cell? A cell is composed of organelles, which considered as an internal organ of the cell. Just like human body made up of various organs like kidney, heart, liver, lungs, likewise cell having different organelles which perform their own functions, and these organelles present within the cytoplasm. Below the table of the organelles found in the basic animal cell, which we will be using our temple for the discussion. First, nucleus. Nucleus was discovered by Robert Brown in 1831. It is usually in different shapes like spherical, oval, and sometimes rod-like. These nucleus absent in few cells like red blood cells and some of the plant cells. Nucleus is completely membrane-bound organelle, uh, which have heredity material in the form of chromosome. These are the structural uh, part of the nucleus: uh, nuclear envelope, nuclear membrane, nucleus, chromosomes, and nucleoplasm. And the total average diameter of the nucleus is 6 micron, which occupy about 10% of the cell volume. Nuclear envelope or nuclear membrane, the double layer membrane, includes nucleus of the eukaryotic cell. The nuclear envelope possesses pores which allow the passage of material in and out of the nucleus. Likewise, nuclear membrane, also known as karyotheca or nucleolema, which is phospholipid bilayer, and inside the nucleus there is a rounded body which is called nucleus, which occupy 25% of the volume of nucleus, which is made up of protein and RNA. Chromosomes are thread-like structure found within the nuclei, both animal plant cells. They are made up of protein and molecule of DNA. Nucleoplasm, which is also called karyoplasm, is a type of protoplasm that made up of mostly of water and different substances. Function of nucleus. Nucleus control the cell activity. It stores hereditary information in the form of DNA. It makes RNA and protein, which makes ribosomes within the nucleus. Second is endoplasmic reticulum. These are the membrane-bound channel which run from the nuclear membrane up to the plasma membrane. These member of the endoplasmic reticulum are found contain different type of enzymes which play various synthetic activities. The physical structure of endoplasmic reticulum consists cisternae, tubules, and vesicles. Cisternae are long, flattened sac-like structure which is about 40 to 50 micron, or it arranged parallel. Tubule, the tubular branch structure forming the reticular system, whereas the vesicles are oval membrane-bound vesicular structure having a diameter of 25 to 50 micron. There are two types of endoplasmic reticulum: smooth endoplasmic reticulum and rough endoplasmic reticulum. Smooth termed is uh, rough termed is it means it coated with the ribosomes, whereas smooth lack ribosome on their surface. Function of endoplasmic reticulum. Its main function is to support the framework of the cell. It also serves pathway or connecting link between the nuclei and cytoplasm, or due to the presence of ribosome, they make proteins, or they also synthesize lipids. Golgi complex, also known as Golgi body. These are membrane-bound structures or organelles which found which are sac-like structure or run parallel from nuclear membrane to the nuclear membrane. And they have a three parts: cis, medial, and trans. Cis, which is nearest to the endoplasmic reticulum, whereas trans farthest from the endoplasmic reticulum, whereas the center part between cis and trans is medial. Cisternae or flattened sac of the Golgi complex uh, is a membrane-bound spaces which various material secretion may occur, or the group of these Golgi complex are that's why termed as Golgi apparatus. Tubules are complex array of associated vesicles and tubules 30 to 50 nanometer in diameter. Uh, Third one is vesicle. Vesicles are three types: transitional, secretory, and Clathrin. Function of Golgi complex. Golgi complex perform various functions. It is responsible for the transportation, modifying, packaging of protein and distribution of the protein and lipids into the vessels and to the targeted destination. They also involve in the transport of lipid molecule around the cell, and these Golgi are involved in the sulfation process of certain molecules. Lysosomes are membrane-bound, dense granular structure containing the hydrolytic enzyme which is responsible for the intra and extracellular digestion. Lysosomes are without any characteristic shape of structure. They are pleomorphic. They are most globular organelle in the appearance. It is about 2 to 5 micron in size, and it is made up of lipoprotein. These lysosomes are single membrane, and these are lysosomal associated membrane protein, which is SLAMP, and the lysosomal integral protein LIMP. It considered about 50% of the all lysosomal membrane protein. Lysosomes functions are heterophagy, uh, programmed cell death, autophagy, autolysis, fertilization, and chromosomal damage. Means, as we said, that they do both type of functioning, extracellular and intracellular digestion. Sometimes they secrete enzymes and they eat their own membrane. Such type of term is known as autophagy or autolysis, or one of the function that is called apoptosis or programmed cell death, that is also occurred due to the lysosomal activity. And heterophagy means they destroy the extracellular material or to eat the extracellular substances. Paroxysms. Paroxysms are membrane-bound organelles occurring in the cytoplasm of almost all eukaryotic cells. These are the particles of about 100 to 500 nanometer in diameter. These are lipid bilayer membrane and regulate enter and exit the paroxysm. Inside a dense matrix urate oxidized crystalline core, there are at least 32 known proximal proteins are called peroxins. Function of peroxisome. Enzyme present in the peroxisome both lead to the production of elimination of uh, hydrogen peroxide, which is reactive oxygen species, synthesis of cholesterol and alcohol occurs in both endoplasmic reticulum as well as in peroxisomes. Mitochondrial, it is a double membrane bound structure present within the eukaryotic cell. These are the structure of the mitochondria. They have an outer membrane, inner membrane, intermembrane, cristae, and matrix. Outer membrane, which covers the mitochondria, or it is a protein structured, which is called porin. Inner membrane, which is free, permeable, one oxygen to oxygen, carbon dioxide, and water contain complex of the ATP synthesis and complex and transport protein. Intermembrane space matrix, the space between the inner membrane and the outer membrane, that is called intermembrane. Crystal is a folding within the mitochondrial membrane. Mitochondria function is the most important function. Is mitochondria is to produce energy. Mitochondria help in the maintaining of proper concentration of calcium ions, whereas the mitochondria also help in the building certain part of the blood and hormones. These mitochondria also play an important role in the programmed cell death. Cytoskeleton is a complex network of filaments and microtubules which form the structural framework of the known cytoskeleton. These are the part of the cytoskeleton: intermediate filament, actin filament, and microtubules. Intermediate filament, the diameter is about 10 nanometer or tensile in length, or it is made up of intermediate protein of two antiparallel dimer. Microtubules are made up of tubulin protein, which is 25 nanometer in diameter. Most of the cilia and flagella are the locomotory organelles made up of microtubules. Microfilaments are the thinnest part of the cytoskeleton and they're made up of actin, protein that is actually the most abundant protein in most eukaryotic cell. Actin is both flexible and strong, make it useful protein in the cell movement. In the heart, concentration is mediated through an actin-myosin system. These are the function of the cytoskeleton. It helps in the cell maintain its shape and give support to the cell. A variety of cellular organelles are held in a place by the cytoskeleton. The type of the intracellular movement supposed to cytoskeleton. The cytoskeleton assists in a transportation of communication. These are the references. Thank you.